Hi, we are subgroup 2 from group 2. This is our video for practical 2A spectrophotometry sugar analysis using the 3,5-dinitrosylicyclic acid (TNS) method, and practical 2B determination of sugar in sample using the 3,5-dinitrosylicyclic acid (TNS) method.
Hi, my name is Jolene Yoiting and my matrix number is 99321.
Hi, my name is Alexa Shelina Jacob and my matrix number is 98785.
Hi, my name is Alicia Emily with matrix number 98782.
My name is Ira Wahyudi binti Abdul Hadi with matrix number 99162.
The objective of this experiment are to determine the concentration of reducing sugar in a sample by using the DNS method. Next, to construct a glucose calibration curve by measuring the absorbance of non-glucose concentration at 540 nanometer using a spectrophotometer. Lastly, to apply the calibration curve to estimate the concentration of an unknown sugar sample based on the absorbance value. The material used for this experiment firstly is TNS reagent. It reacts with reducing sugar when heated to produce a reddish-brown color for measurement. Next, Rochelle salt to stabilize the final reddish-brown color to ensure an accurate reading. Cuvette, a transparent container that holds the final solution for analysis in a spectrophotometer. Hot plate provide the necessary heat to drive the chemical reaction between the sugar and DNS reagent. Glass beaker used for placing the test tube during boiling process. Distilled water acts as a solvent for creating standard solution and used as a plain liquid. Test tube and rod serve as the cell for the reaction and are held by rack for organization and safety. Micropipette and tips used to accurately measure and transfer precise small volume of liquid. Selected drink sample, the substance being analyzed to determine its sugar content.
This is our results for the experiment. The average and the standard deviation of absorbance at 575 nanometer were calculated for test tube 1 until test tube 6.
In conclusion, we successfully quantified reducing sugar using the DNS method. We show a strong linear relationship between glucose concentration and absorbance, consistent with the clear line below, R2 equal to 0.9963. Duplicate readings were consistent, reflecting accurate handling and execution. In the twin sample analysis, absorbance was measured at 575 nanometer using a spectrophotometer. Test tube 1A and 1B recorded the highest absorbance value, while test tube 6A and 6B show no absorbance, indicating a decrease in reducing sugar concentration from tube 1 to tube 6. The absorbance value aligned with the glucose calibration curve, confirming that higher absorbance corresponds to higher sugar content in line with the Beer-Lambert principle. In conclusion, we successfully quantified reducing sugar using the DNS method. We show a strong linear relationship between glucose concentration and absorbance, consistent with the Beer-Lambert law, R2 equal to 0.9963.
This is our group reflection. In this experiment, we learned how to determine glucose concentration using the DNS method. We gain a deeper understanding of how reducing sugar react with DNS to form a color complex and how this reaction is quantified using spectrophotometry. We also practice preparing a glucose standard curve and interpreting the linear relationship between glucose concentration and absorbance. Additionally, we improve our skill in using micropipettes accurately, handling hot water bath, and using the spectrophotometer for absorbance measurements.